So let's talk about the structure of DNA. So DNA is a macromolecule or a polymer. So that means that it's a very large molecule and it's made of smaller subunits. So the subunit is called a nucleotide. So that's what we call the monomer. Mono meaning one. So that's the subunit. When we put them all together, we have what's called a nucleic acid. So the nucleic acid is the polymer, otherwise known as a macromolecule. Now, nucleic acids, we've actually got two different types. We have got DNA and we've got RNA. A couple of really important differences. Firstly, DNA has got two strands, whereas RNA has got one strand. A couple of other differences, they have a different sugar. So I'm going to talk to you about the actual structure of a nucleotide in a second, but one of the parts of it is a sugar. And in DNA, it's called deoxyribose sugar. Okay, so DNA, the D in DNA stands for deoxyribo, nucleic acid. RNA has a different type of sugar called ribose sugar. So its RNA is ribonucleic acid. One more thing, another part of our nucleotide is called the base, and we have different types of bases. One of them is different in RNA and DNA. I'm going to show you the basic structure of a nucleotide, and then we're going to put it together to make DNA. So this is what a nucleotide looks like. First of all, we've got our sugar base. Okay, so the O here is for oxygen, and each of these corners is a carbon. And then we have another carbon here. Right, now, coming off this, well, first of all, what I'm going to do is I'm going to show you how we number the carbons. So the way we number carbons in organic chemistry is in a clockwise direction, and we start with the carbon that's closest to the oxygen. So we've got oxygen here, so this is carbon 1, carbon 2, 3, 4, and this is carbon 5 here. Coming off carbon 1 is where we have our base. So our nitrogenous base. And we have a number of different types of bases. Then where we've got our carbon, or our carbon 5, hanging off that is the third part of our molecule, which is the phosphate. So we've got three parts of our nucleotide. We have got our sugar, we have got our base, and we've got our phosphate. So that is a nucleotide. Now, next thing I need to show you is how we put these together to form a nucleic acid. Okay, so this is our nucleotide, and remember it's got the three parts. You've got your sugar ring, you've got your base, and the phosphate. And we also numbered the carbons. So this is carbon 1, and the base comes off carbon 1. Carbon 2, carbon 3, carbon 4, and that's carbon 5 there at the bend, and the phosphate comes off carbon 5. Right, so we're going to add our next nucleotide. Now, the next nucleotide is going to bond to the third carbon of this nucleotide here. So our phosphate of our next nucleotide joins onto or bonds to the third carbon. So now we can go ahead and just replicate as best I can. Okay, so then we've got our second nucleotide, and of course then our next one is going to come off carbon 3, then we'll have our phosphate. I've just labelled a couple of our carbons here. Down here, this is of course the 1, 2, this is the third carbon. And then up on this one, 1, 2, 3, 4, that's our 5th carbon. So not only can we label or name the carbons in our individual nucleotides, we can actually label the whole DNA strand by this information as well. So we call this end of our DNA the 3' end, and this end we call the 5' end. So the actual DNA strand has directionality as well. So 3', that's what we mean, this prime, so 3' end and the 5' end. Now, remember how DNA has got 2 strands. So we need to go ahead and draw our other strand of DNA here. Now, what we say is the other strand of DNA is anti-parallel. Anti-parallel. So imagine it's just like a roadway where you've got cars going in this direction and cars going in this direction. So they're running parallel, but they're going in opposite directions. So we say that's anti-parallel. Our DNA is exactly the same. It's anti-parallel, whereas this one runs from 3' to 5'. This one's going to run from 5' to 3'. So in other words, it's inverted. So it's a little bit harder for me to draw. Normally what I do is I turn the page over, which I can't do here. So I'm just going to take my time and go ahead and try and draw it, and then I'll explain it to you. As you can see, it's a little tricky to draw, so obviously they all should be the same size, and mine isn't quite the same size. Let's go ahead and label this strand. So let's see, if we count these carbons again, that's the O, okay, so that's carbon 1, 2, 3, 4, 5, and if we go down to this one, we've got carbon 1, 2, 3, 4, 5. Okay, so that means that this is our 5' end and this is our 3' end. So as you can see, they're anti-parallel. This one runs from 3' to 5' and this one runs in the opposite direction. So it's anti-parallel. One more thing I need to tell you is our bases join together in the middle with weak hydrogen bonds. Because of course the DNA needs to unzip. So we've got these weak hydrogen bonds that hold the 2 strands together. So you can see that the primary structure of DNA looks like a ladder. We've got 2 strands and they've got these backbones, so we call it a backbone, a sugar phosphate backbone that continually repeats. And in the middle for our rungs, we have our bases. And that continually repeats. Now I said that's the primary structure because it's also got a secondary structure as well, and that is it winds up as a double helix. The primary structure, the secondary structure is a double helix. And that is our DNA structure.